All right, we're going to get started so we can stick on time. So welcome everybody to our second network speaker series for C-DEBI, the Center for Dark Energy Biosphere Investigations. I'm Julie Huber. I'm the Associate Director of C-DEBI, and I'm very happy to introduce Olivia Nigro today. She is a C-DEBI postdoctoral scholar. Olivia has been living in Hawaii for a good time now. She got her PhD there with Greg Stewart. And while she was there, she actually mainly worked on viruses associated with Vibrio. And more recently has started moving into the subsea floor with Greg. She's been a C-DEBI postdoc and is also part of a number of C-DEBI projects. So Olivia, I'm going to turn myself off and hand it over to you. And just for our audience, the way that you ask questions is by typing them in the chat box. Olivia is not going to look at that during her talk. So if you want to make a note there of something you want to ask her, that's fine. But all questions will be happening via the chat at the end of the talk. So take it away, Olivia.
Okay. Hi, can you all hear me? Okay, good. Thank you. Okay, thank you. And thank you, Julie, for that introduction. First and foremost, I would like to acknowledge my collaborators who helped me extensively with this work, including Mike Rapay and Sean Youngbooth, who contributed a lot to the complementary microbial analysis, to the viral analysis that I have done. I would also like to thank C-DEBI for the opportunity to conduct this research as part of my postdoctoral work and the opportunity to speak here today. So today I'm going to present to you data that I have collected on viruses inhabiting basalt-hosted crustal fluids of the Juan de Fuca Ridge flank. Most of the studies investigating viruses in subsea floor environments have considered only sediments and hydrothermal vents. And today I'm going to present to you some of the first data on viruses in the basalt-hosted basement. So the word virus often evokes images of the cold or the flu, childhood illnesses like the measles or chickenpox, or even more serious viral-borne diseases like HIV. However, it is estimated that of the estimated 10 to the 31st viruses on Earth, that the majority infect microbes like the bacteriophage rendered here. In particular, bacterial and archaeal cells. So studying viruses in their natural environments, including the world's oceans, have revealed that some of these viruses defy the conventional paradigm that viruses' role in the environment is solely tied to cellular destruction. As I mentioned, viruses are abundant in marine environments and can have profound effects on the microbial population structure. If we had a volume of surface seawater that was equal to this little plastic square that you see, that is 1 millimeter on each side and is a volume of 1 microliter, there would be about 10,000 viruses present. And the viruses you see in this image are the small green dots, and the larger green dots are microbial cells. In surface seawater and throughout the water column, viruses outnumber microbial cells by a ratio of about 10 to 1. And this is throughout the water column, at depth, and oftentimes in the sediments as well. The process of lytic infection, which is one of the viral life strategies that you see here at the top of this slide, is important in marine environments because in the lytic pathway, a virus infects the cell and then eventually lyses and kills the cell. And this is important for several reasons. First of all, because this reduces the number of the target cells in the environment. Secondly, during this infection process, viruses are capable of enhancing specific aspects of cellular metabolism. For example, in the case of viruses that infect cyanobacteria, they are capable of increasing the rate of cellular photosynthesis, which in turn increases the amount of carbon fixed in the environment. Similarly, in hydrothermal vent environments, it was recently reported that viruses encode genes for sulfur oxidation, which is a widespread metabolic strategy in these environments. These findings suggest that viruses may be intimately involved in sulfur cycling in these environments. And finally, the lysis of these cells returns the host cell carbon and nutrients that were previously not available to microbes to become bioavailable again. And this ecologically significant process is known as the viral shunt of the microbial loop. However, the lytic cycle is not the only important pathway. The other important life history of viruses in marine environments is the lysogenic pathway. And in this pathway, a bacteriophage, again, enters the host cell, but in this case, the bacteriophage DNA or archaeophage integrates into the host chromosome, and it's now called a prophage. So this little piece represents the microbial DNA, and it's now called a prophage when it's integrated. So it stays integrated in the cell until an environmental trigger causes it to be released. And the lysogenic pathway has implications for horizontal gene transfer. Viruses can pass genes from donor to recipient cells in a process called transduction. And viruses themselves can also encode genes that are transferred between host cells in a process called lysogenic conversion. So these are the basic life strategies of viruses in the marine ecosystem. So there are some challenges associated with studying viral ecology, and a lot of these stem from the fact that viruses are very small, and following, they have a very small amount of DNA per virion. So when we're doing work with viruses in the marine environment, we have the challenge of obtaining a large enough sample to be able to measure viruses, as well as to have enough DNA to do molecular analyses. Another issue that's an issue in all of microbial ecology is that when you do metagenomic analysis on natural populations, this causes a disconnect between the physical properties of, in this case, the viruses and the genomic information. So the actual process of creating a metagenome destroys the phenotypic information about the viruses. And the last challenge associated with viral ecology is that it's very difficult to link viruses to their hosts. So when you make a viral metagenome, you're essentially collecting all the viruses in the environment, but this doesn't really tell you what viruses, which host the viruses are infecting. And this is often the question of viral ecology, is how are the viruses affecting the microbes in the environment? And there have been several recent advancements to try to make it easier to make this connection, including this technique called phagefish, which uses a in situ hybridization technique to try to link viruses and their hosts together. However, this is not ready to be used in a natural environment. It's more for cultures. So this is still a problem that exists in viral ecology. Okay, so what do we know about viruses in the basement? Well, as I mentioned earlier, there have been no reports on viral communities in oceanic basement fluids. However, there have been studies on viral communities in hydrothermal vents and in deep sediments. And coincidentally, both of the authors of these papers were previous speakers in this seminar series. So good job, C-DEBI, on supporting the viral people. So since no reports have been, or viruses have not been described in these environments, many of our questions are generally first order questions. Are viruses present in the basaltic basement? And you can probably guess from the fact that I'm doing a 30-minute talk on viruses in the basement that we did find viruses in this environment. But how abundant are these viruses? What do the morphologies look like? And how are they molecularly characterized? But also, we want to know how significant the ecological role of the viruses is in this environment. And also, can we see difference in the ecological roles of viruses, for example, that infect archaea and that infect bacteria? So these are some of my questions going into this study. So now I'm going to tell you a little bit about the anatomy of the seafloor and the environment that we're working in. As most of us learn early on, about 70% of their surface is covered by the ocean. And in our minds, we can picture the ocean gyres churning, moving nutrients and planktonic organisms throughout the water column. But surprisingly, seawater circulation does not stop at the seafloor. Despite thick layers of sediment that cover much of the ridge flanks and basins, seawater can enter the basement basalt at unsedimented or lightly sedimented outcrops. And contrary to the sediments, which serve to restrict fluid flow with the upper water column, the basalt is actually quite porous, and water percolates through pores and cracks and fissures and is able to stay in this environment for a few to thousands of years. And the crustal fluids eventually reemerge with varying degrees of geothermal alteration and biochemical alteration at distant outcroppings. So the basement aquifer illustrated here includes all the basement fluids, and this is equivalent to about 2% of the ocean's volume. And the fluid flux through the ocean basement recycles the full volume of the global ocean every 100,000 to million years. Additionally, annually, the volume of fluids that moves through the crust is equal to the global annual flux of riverine input. Okay, so what do we know about the microbes in this environment? So early studies were driven in part by the notion that the ocean basement and the microorganisms that inhabit it are a global scale biochemical reactor, altering seawater and influencing the chemical equilibrium of the entire ocean. So early studies by Jim Cowan et al. And others using basement fluids from the eastern flank of the Juan de Fuca Ridge, which is also my sampling site, indicated that microbes are present in these environments and that the microbial community is likely dominated by sulfate reducers, unique chemolithoautotrophs, and thermophilic archaea. And more recent studies in this environment have shown that the microbes in the basement fluids, which are represented by, in this image, by the red, brown, and yellow groups, are distinct from the microbes that are found in seawater, which are these blue groups, and sediments, which is this darker brown group down here. Okay, so you can imagine that sampling the deep subsurface environment is likely difficult. So there are different types of fluids that can be sampled, of different samples you can collect. So you can collect natural fluid outflows from hydrothermal vents, which has been previously done by Rika Anderson and others. You can collect sediment cores to look at the viruses or microbes in the sediments. However, we are looking specifically at the basalt hosted fluids. And in order to do that, we needed special apparatuses called corks. And corks is an acronym for Circulation Obviation Retrofit Kit. And what corks are they are casings and fluid delivery lines that are retrofitted to fit IODP boreholes. We access these using ROV Jason or HOV Alvin, and there have been 3 generations of corks, and each iteration of cork gets better at delivering pristine, unaltered basement fluids. So corks are located worldwide, but I focused on the corks on the Juan de Fuca Ridge flank. And this is an active spreading center, which is about a day's steam from the coast of Oregon and Washington. And the data that I'll be showing today comes from 3 cruises to the corks in 2011, 2013, and 2014. And all the data is from 3rd generation corks, U1362A and U1362B. These observatories are located below 2,700 meters of seawater, and they were made by drilling through 250 meters of sediment and then another 100 to 300 meters into the basaltic crust. The geochemistry of this environment is dominated by cold bottom seawater entering the basement at unsedimented and thinly sedimented seamounts. Over here, the water is heated up during circulation, and basement fluid temperatures increase as the water moves away from the recharge area. And as the fluid travels from the spreading center to the cork, the chemistry is significantly changed. The waters become anoxic and depleted in nitrate. DOC is relatively low, around 10 micromolar, and fluids become enriched in methane, ammonia, and hydrogen. And when the fluids are discharged out of the cork, they're around 65 degrees Celsius. It is obviously important for us to be able to determine how pristine the samples are that we obtain from the corks. And this is easily done. They're easily distinguishable from seawater based on geochemistry. So in this image, you see the separation by magnesium concentration, and seawater is way up here. And then you can see as these colors get warmer, we are moving towards newer second and third generation corks. So these are the earlier corks, and moving down, we're getting to newer corks. So you can see over the years, we have improved our ability to collect pristine waters from the corks. Okay, so as I mentioned before, there are considerations when doing work for viral studies. And the major consideration is that we need a large volume of water. And so we have two methods that we use to collect water. The first is that we use a geomicrobe sled, which is a device that is sent down to the bottom and with the use of alvin is attached to the cork, and fluid is collected in bags and then brought shipboard for processing. However, on our 2014 cruise, we deployed a filter in situ in order to do passive filtration so that we could obtain a larger volume. And this resulted in 10,000 liters of water filtered, and we were able to recover 8 times 10 to the 10th viruses on our tangential flow filtration unit. So I'm going to just show you a little video of some of these sub-ops at the cork. So here is the arm of Jason, and it's hanging, actually it's Alvin, sorry, and it's hanging our sampling package onto the cork, and it's going to first attach a line directly to the cork, and then the line is going to be flushed for a while, for 10 minutes, I think. So there's the Alvin putting in the connector and turning it on. You can see this shimmer of water coming out, of the warm water coming out of the cork. So after it purges for 10 minutes, then we hook it up to our filter, and the filtration begins, and there's that shimmer of water again. And our filtration rate was about 1 liter per 50 seconds. Okay, so here are some of my results, starting with microscopy. So abundances were determined by counting Cybro gold stained virus-like particles from samples of 50 to 75 mils of whole deep subsurface fluid filtered onto a 0.02 micron anodisk filter. And what we found was that on average, viral abundance was 1.1 times 10 to the 5th. And we did see the same virus to cell ratio that has been previously observed of around 10. And in bottom seawater, the viruses and microbes were both an order of magnitude higher than in the crustal fluids. So the next step was to look at the viral morphologies in the transmission electron microscope. So I'm going to show you some known viral morphologies first to try to set the stage for what we found. So what you see here in this image are tailed and non-tailed bacteriophages. And in this paper by Brum et al., Jen Brum looked at a large number of samples as part of the Terra Ocean project, from surface seawater down to about 400 meters. And in these samples, she reported only seeing these 4 morphologies. These tailed viruses are known collectively as Caudovirales. And they're just little icosahedral head with either a small tail, a medium tail, or this long flexible tail. And then this is a non-tailed virus. And this similar viral morphologies were observed in the sediments in a study by Engelhardt et al. In 2014. So they saw these similar viral morphologies of the non-tailed viruses and these different tailed groups. When you look at the known archaeal viruses, however, they have vastly different morphologies. So these are some viruses that infect thermophilic archaea, and they have these lemon-shaped, club-shaped type viruses, and also different types of filamentous viruses, stiff rods and flexible rods, as well as these globulo types of viruses. So they're vastly different than the typical bacteriophages. So these are the viral morphologies that I observed in our Juan de Fuca Ridge flank samples. So you can see that we did have some of these typical tailed bacteriophages, but then we also see these archaeal type morphologies. Here's a lemon-shaped virus. This looks like a globulo type. These two are both the rod type. This looks like the club type. And then this type is one that we observed several times that we have not seen described anywhere else. And I would like to note that many of these virus morphologies have not been directly observed in marine environments before. They are commonly isolated from terrestrial hot springs, and some have been isolated in enrichment cultures from vents and from hypersaline lakes, but they have not been directly observed in the marine environment previously. Okay, so onto my metagenomic analysis. So for our metagenomic analysis, we had two targets for viruses. And the first is free viruses. And we can look at the free viruses by looking at the viral metagenomes that we made. So this is the fraction that's less than 0.2, and then what's collected on a tangential flow 100 kilodalton filter. We can also look at viruses that are associated with cells, and these are the viruses that we find in the greater than 0.2 micron fraction. So the types of viruses that we would likely find there are replicating viruses, so viruses that are currently in the process of replicating within a cell. Lysogenic viruses that I described earlier that are integrated into the host chromosome. Another way that I'll discuss in depth later is viruses can be can be shown as part of the CRISPR-Cas system. And additionally, we also do get free viruses, and these are large viruses that are greater than 0.2 micron and get caught on the filter. Okay, so I'm going to first talk about my viral metagenomes. So in preparation to make our viral metagenomes, we decided to try something that is non-standard for making viral metagenomes. So in order to decrease the complexity of the viral metagenomes, we, before sequencing, physically fractionated the intact viral particles. And this is also important because, this is also important because it will retain some phenotypic information about the viral particles. So what you see here is our cesium chloride buoyant density gradient. And the bands that you see here are actually bands of viral particles. So we take this cesium chloride gradient and we spin it for several days in an ultracentrifuge. And viruses band out with the density gradient that they are the same as. So then we took this density gradient and we sipped off fractions from the density gradient, and we ended up with 17 fractions. And from these 17 fractions, we made 17 metagenomes. And these were done in the standard way. After metagenomes were made, we assembled using IDBA. And so first I'm going to show you how well our fractionation worked. So this is the buoyant density fraction. So if you can imagine that column that I just showed you, but turned on its side. So these are the buoyant densities. And graphed across this are the reads mapped to representative viral homologs. So in blue are the archaeal viruses, and throughout the rest of this talk, archaeal viruses will be in blue. bacterial viruses will be in red. So remember, there are these oddly shaped viruses, and then the bacterial viruses are these this tailed group. So what we found is that in these lower density fractions, we were actually able to enrich for archaeal viral sequences. And I went back and looked up the buoyant density for archaeoviruses and Caudovirales, which these tailed viruses that dominated our bacterial signal, and plotted here are the actual buoyant densities of the known representatives of these viral groups. So from these metagenomes, we were able to obtain 34 closed unique viral genomes and 110 linear scaffolds with at least 3 ORFs and 1 viral hallmark gene. And so this is a high number of assembled viral genomes, and hopefully that is a representation of the fractionation, which is essentially like a pre-binning process of putting similar viruses together. Okay, so moving on to looking at the community composition of the viruses. So again, here is my buoyant density fraction on the x-axis, and on this axis, it's the percent of representative viral homologs that mapped to of reads that map to the representative viral homologs. So you can see that in these lower buoyant density fractions that the sequences were dominated by Rudiviridae, which is a type of archaeovirus. And then in these higher density gradients, we have Poto, Myo, and Cyphoviruses, and these are the groups that make up the Caudovirales. But of course, we're interested in comparing these metagenomes to other environments. So what you see in this bar is the Juan de Fuca ridge flank microbial metagenome and the distribution of archaeal sequences and bacterial sequences. And this is viral metagenomes from a bunch of different environments. But first, here is the Juan de Fuca ridge flank. So we have about 55% archaeovirus signal and around 40% bacterial. And this is largely different from what you see in open ocean environments, such as the surface Pacific. Even in the deep Pacific, we don't see this high abundance of archaeoviruses. The only place where we saw a high abundance of archaeoviruses was in hot springs from Yellowstone. And even in vent environments, they seem to be dominated by bacteria. So how does this compare when looking at other deep subsurface omic studies? So now this is the same plot, just broken down to the viral family level. So here's my sample from the Juan de Fuca ridge flank, and here's Yellowstone and the other ocean environments. So you can see this blue part is Rudiviruses. So even though the Yellowstone sample did have archaeoviruses, they don't appear to be the same, dominated by the same type of archaeoviruses as you see in Juan de Fuca. And again, the open ocean ones are dominated by the Caudovirales. So in this report from Engelhardt 2015, they did a similar kind of analysis looking at RHV's representative viral homologs in the sediment. So this is depth into the sediment. And they did see some archaeovirus sequences, particularly in this top fraction. They saw some Lipothrix-viridae, which is a rod-shaped virus. But the dominant type that we had, the Rudivirus, they only see a very small portion of their reads are similar to that type of virus. And Enrica Anderson's study from hydrothermal vents, she found that the archaeovirus signal was very, very low, rare, if present at all, in her vent samples. Okay, so the next way that we can identify viruses in our metagenomes is by looking for prophages in the microbial metagenomes. So we did this using the program VirSorter. And using this program, which looks for prophages in microbial metagenomes, we were able to detect 50 prophages in 1362A and B. I'm just going to show you what that looks like. So here is one of our larger scaffolds from the microbial metagenome. And this portion right here, all these white genes are what was identified by VirSorter as a prophage. And then this down here is actually the 16S and 23S gene of this microbial fragment. So we were able to use that to identify what group this prophage infected, and it's actually a novel lineage in the terrestrial hot springs group. So looking at this, the genome of this prophage more in depth, so this is the only viral gene that was identified by IMG. So I re-annotated the genome using the viral subset of the non-redundant database, and I was able to identify 6 additional viral proteins, including a phage integrase right here on the flanking end of the prophage. These genes that you see, they're all annotated as other or unidentified. And this is what makes up the large portion of viral metagenomes that are unidentified and microbial. So using methods like this, hopefully it will aid in improving metagenomic annotations. And so then I looked at these, the hits that I was able, the genes that I was able to identify, and the most common hit was to Acidianus filamentis, which is a virus that infects a hypothermophilic Crenarchaeota. So we were not so fortunate to have every prophage on a scaffold of the 16S RNA gene, but as part of a separate but related project, Sean Youngluth and Mike Rapay have been working to refine genomic bins from these microbial metagenomes. In total, 99 genomic bins have been built and assigned taxonomic identifications. We were able to determine which genomic bin each prophage-containing scaffold is assigned to. And this is a breakdown of the 33 bins that the 50 prophages map to. So archaeal bins are shown in blue and bacterial bins are shown in red. The number of the genomic bin is noted here, but more importantly, here is the taxonomic identification of the genomic bin. So we found that there were many prophages in the Archaeoglobus and the related Archaeoglobi bins, which we think is a novel lineage. We think these are novel lineages of these archaea. And they also had several prophages in each bin. And this is also the case with Thermococcus and others. Prophages were more common in archaeal bins than bacterial bins. So the outer ring in this image is the 99 bins separated as archaea, of which is 45%, and bacteria, which is 55%. And the inner ring shows which domain each prophage mapped to. You can see that archaeal prophage abundance is overrepresented compared to the bacterial. And a simple way to look at this is the percentage of each bin type that has at least one prophage. In this case, around 45% of the archaeal and 25% of the bacterial had at least one prophage. And these numbers, I think, are reasonable. Another study that was done on tens of thousands of microbial metagenomes found that around 30% of microbial genomes harbor a prophage. Okay, so the final way that, that we investigated viruses in this environment was to link the viruses to the host using CRISPR systems. And CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And basically, the CRISPR system is a primitive immune system in prokaryotic cells. So when a virus infects a cell, the viral DNA enters the cell and then it is cleaved. And that DNA is then inserted into a CRISPR array. So looking at this array let's look at this array a little bit closer because there's portions of it that I talk about, so it will be good for you to understand. So this portion here is the microbially encoded Cas genes, which stands for CRISPR-associated proteins. And then here in black are microbially encoded direct repeats. So these parts are both encoded by the microbe. And then interspaced between the direct repeats are the virally encoded spacers. So these are the pieces of the virus that were chopped up and then added sequentially. So this little CRISPR cassette is actually a record of past virus infection. The only real limitation of using this to investigate past infections is that these spacer sequences are short, upwards of 60 base pairs. Okay, so in order to use this to investigate viruses in the deep subsurface, we first identified all the CRISPR cassettes in the microbial metagenome, and we did this using a program called CRAS, and this inspects raw metagenomic reads for CRISPR elements. And the CRAS program assigns CRISPR groups based on the microbially encoded direct repeat portion of the CRISPR gene. In order to make taxonomic identifications of CRISPR flagged reads, CRISPR groups were then recruited against our microbial genomic bins. And we found that CRISPR genes were abundant in the metagenome, and they were also present in a broad range of taxa, as you can see here. Additionally, we found that there's a cohesiveness between the groups and the genomic bins. So for example, all these CRISPR groups here only infect archaeal groups. Okay, so this is a rank abundance plot of the 16S RNA from the assembled 1362A metagenome. So this is the most abundant species, microbial species in the 1362A metagenome according to 16S RNA. And wherever you see a plus, that is where we had a CRISPR element. And you can see here that it's widely distributed through the abundant taxa. And this is suggestive that viral infection is an important process in the deep subsurface environment, since microbes are maintaining DNA to encode elements to defend against viral infection. Okay, and then so the ultimate goal is to link the spacers back to their microbial hosts. So this analysis allows us to do this. So what I did was I then took the spacers that were recovered, the short spacer sequence that was recovered from the microbial metagenome, and that was recruited to the viral metagenome, to our viral metagenomes, which had larger pieces that then we could investigate. And so I only looked at spacers that had 100% similarity to the viral metagenomes, and in total, there were 3,000 spacers from the microbial metagenome that matched contigs from our viral metagenome. And so then we can look at the groups that these infect. So this is that same heat map that I just showed, but turned on its side. So we can look at these particular groups. So for example, group 34, which we had the most similarity between spacers and the viruses, this did not match up with any of our genomic bins. So that means that either we don't have a genomic bin for, well, likely it means that we don't have a genomic bin for this particular group. However, if you look at group 25, you can see that all of these spacers fell within the marine benthic group E. So now when I look at these viral sequences in my viral metagenome, I can say with some confidence that they likely infect marine benthic group E. And then there were other groups, such as this group 3, which were more broadly distributed among the genomic bins, which could mean a couple things. It could mean that our bins need to be more refined, or it could mean that these maybe have a broader host range. And then additionally, I plotted these, the distribution of the hits between the spacers to the viral metagenome. And I saw a similar distribution of the hits that I saw in the fractionated genome. So this group, which hits an archaea, is clustered in the lower fractions of my fractionated genome. This group 34, which we don't know what it infects, is grouping in the larger range. Okay, so the summarized findings from my study, we found that viruses are present in basalt-hosted crustal fluids, and they are morphologically diverse when compared to both seawater and sediments. Physical fractionation of intact viral particles resulted in enrichment of archaeal viral-like sequences in the low-density fractions. The viral assemblage in crustal fluids is genetically distinct from other sequence communities, and analysis of microbial CRISPRs indicates that viruses are likely to be actively infecting microbial populations, and these CRISPRs can then be used to identify broad taxonomic host groups infected by specific viral genomes. Okay, I would like to acknowledge C-DEBI for all of their support over the past few years. I would like to acknowledge my C-DEBI collaborators here at UH, which started with Jim Cowan, my postdoctoral mentor, Greg Stewart, our collaborators, Mike Rapay and Sean Youngbluth. I would also like to thank our Wanda Fluker Ridge collaborators, Andy Fisher, Jeff Wheat, Brian Glazer, and Beth Orcutt. Seymour and Scope, they give me a home here at University of Hawaii. And our funding agencies, the captain and crew of the RV Atlantis and the HOV Alvin and ROV teams and the electron microscopy facility here at the University of Hawaii. Thank you, and I'd be happy to take any questions. Hi. Okay. Okay, so I think I'm just going to start digging into these questions. Okay, so the first is from Rika Anderson, and she says, hi, it's really interesting that you saw more prophage in archaea than bacteria, if I understood correctly. Yes, that's correct. Any ideas if archaea are just more likely to harbor prophage than bacteria, or if they're easier to detect? How does incidence of lysogeny lysis in ridge flanks compare to other regions in the subsurface? Okay, so I haven't seen any other studies in the subsurface environment looking at the distribution of prophages, particularly in bacterial and archaeal groups. I mentioned that study by Simon Rue, where they looked at a huge amount of microbial genomes and found that they were present in 30%, but they did not break this down into bacteria and archaea. And I actually emailed him to find out if they did see any differences. And what he told me was that the abundance of archaea isolates that they used in that study was so low that he didn't feel like it was statistically significant enough to report those findings. So I think that that question is pretty wide open. As for the reason why archaea are harboring more prophage than bacteria, if that is actually true, there are some papers that describe archaeal virus life strategies, and they differ from those traditional lysogenic lytic pathways in that sometimes they have like a chronic infection where the virus infects the host genome and then it stably buds off over time. And it was thought that for a while that these were kind of stable relationships between the host and the virus. But I think that's being revisited because it has been shown that the viruses actually do significant damage to the host cell. So it's not like a mutualism, it's still kind of a parasitic type relationship. So the hypothesis as to why that strategy evolved in archaea, I think, hinges on the idea that they live in extreme environments and that the cell abundance tends to be lower, and so the contact rate of viruses to their hosts is likely lower in those environments. So a lysogenic strategy would be beneficial, but that you would expect that that same evolutionary pressure would be there for bacteria as well. So I don't know if that completely explains it, but does that answer your question? Okay, so hopefully that answers your question, Rika. Okay. Okay, my next question is, out of all the sequences assembled, what percentage is unclassified? Could we be missing out on viral diversity that are unclassified? Also, why are some of the microbial genome bins not labeled? Okay, so yeah, I didn't here. Can I still share my screen? Is my screen still being shared? I didn't show this just in the interest of time, but in my viral metagenomes, about 83% is unknown and only 2% were identified as viral. So what I showed was actually just representing 2% of the sequences. So yes, there's a lot of viral dark matter that we are not able to classify. So we're definitely missing out on viral diversity, but the fact that we can assemble genomes, that's going to help us to identify some of this viral dark matter, because when I look at those assembled genomes, only a few of them were actually annotated as viral. So only a few of those are contributing to that 2%. And the large majority of those sequences are not contributing to the known portion. So since those have assembled, and I'm certain that they are actually viruses, then that's going to help us to observe and annotate more of the viral diversity than we have been able to do in the past. And then programs like VirSorter, where you can pull those genomes out of microbial genomes that are not annotated at all as viral, that will help also to build up the database to identify viral sequences. Let me see, there's one more question in there. Also, why are some of the microbial genome bins not labeled? So these genomic bins are in a process of refinement. They're getting better and better all the time, but some of them have not been, although those sequences have similar tetranucleotide frequencies and similar read depth, we have not been able to identify which taxonomic group they belong to. And that project is being worked on by Sean Youngbluth. That's his specialty. Okay, so the next question from Julie from USC. How do you know that some of the oddly shaped viruses are actually viruses, especially some of the weird archaeal morphologies? That's a really good question. So you may notice that we didn't do quantitative transmission electron microscopy, which is when you go through and you try to count all the viruses of a particular type. And that was exactly for that reason. It's difficult to be certain that all the particles you see are what you think that they are. However, for the archaeal viruses, like for example, the lemon-shaped viruses, oftentimes you can see appendages on the end of them. And they're also in other known strains. So, and also the fact that we see that same shape over and over and over again. If it was just something that we observed once, we would be more skeptical that it was actually a virus. So, so yeah, like for example, the viral type that I said has not been observed before, we would not be able to prove that that is actually a true viral morphology until we were able to isolate it from a microbe or an archaea or a bacteria. So we don't actually know for certain that that's a viral morphology, but it's a putative viral morphology. So I think that's the only one in that group that's kind of borderline. But we did see that one several times throughout several different samples. So that builds up our confidence that what we're seeing is actually a virus. Okay. Have you had the chance to determine if there's much? Okay, so the next question is from Jason. He said, have you had the chance to determine if there is much change in the viral composition across the 3 years you sampled, especially in the free living virus fraction? So if you mean metagenomically, no. The only viral metagenome we have so far is the one from 2014. However, we do have samples that are going to be processed to look at that. So hopefully we will look at that. And then if you mean morphologically, again, we didn't do quantitative TEM, so it's only qualitative at this point, describing the type. So in that way, it's hard to say if there are any changes in the across time. But when we get the other viral metagenomes back, then hopefully we'll be able to answer some of those questions. Okay. From Meng, are there any transcriptomic evidence to confirm the virus target specific archaea or bacteria groups? Not from our study. The data that I showed from Engelhardt was actually from a transcriptome where they saw both archaeal and bacterial groups, but we have not specifically, as of yet, done transcriptomics. Sure. Okay, does anyone else have a question?
Okay. Can you hear me, Olivia?
I can.
Okay. So there is one more question that just came in about the giant viruses.
Okay. So I have not seen any giant viruses, or I do not have any firm evidence of giant viruses in these sequences. Giant viruses typically infect eukaryotic cells, and this environment is either has no eukaryotes or has very low eukaryote abundance. I do see some sequences that may have maybe homologous to giant viruses, but it's not really enough evidence to say that they're present in the environment. Yeah, so I did have intact viruses that were greater than 0.2 micron. So I have what I think are free viral genomes on the greater than 0.2 micron fraction. But largely, I think that those are filamentous archaeal viruses that, when they went through the filter, were oriented in the horizontal manner instead of the vertical manner. So they stuck onto the filter. But I'm definitely interested in giant viruses. Yes, we haven't done, sorry, there's another question. It was, could you also be missing the RNA viruses? Yes, we have not done analysis for RNA viruses yet. That's another thing that hopefully we'll be able to do in the future.
I'm just making sure none of these people typing are questions. I think mostly they're saying good job and thank you. So I will just echo those, Olivia. It's a great talk, and thank you to everyone for joining us. I know for some of you it's breakfast, lunch, and here it's time to head home. So thanks, everyone. Olivia, you might want to just hang out and check out that chat space for a little bit longer.
Okay.
All right. Thank you.